Go ahead and get started here. And yeah, so the music you're hearing, these are actual compositions that a composer scored based on songs he heard mushrooms speak. Right? He has a condition called synesthesia. So it's a neurological condition where, in his case, seeing a mushroom interpreted as sound. And as a composer, I mean, thousands and thousands of compositions, and even within each genus, uh, there were each species has a different song. So anyway, fascinating stuff. So my name is Greg Sanchez. I am past president. I'm gonna kind of introduce things, and I've got a few things with business today, but thank you for coming. This is our July meeting. Anybody been out yet? Anybody find anything yet? Front Range is going to be a little tough, right? So we did do some forays this weekend. I led a foray with some other leaders on Sunday, and we had some luck. Both leads are there. So I don't know if that's encouraging or not, but got to look. Nonetheless, our foray season is well underway. Hopefully you're getting the notifications for signups and more are coming. So we got more Wednesday, Saturday, Sunday, and that's pretty much how the pattern's going to be. And on that note, I want to mention a pretty special person here. Jay, would you raise your hand, or do you want to stand up? Jay is our 4A chair, and so he is really the master behind all the chaos. I think. I mean, I can imagine what kind of emails he's getting, but he's done a great job for us coordinating leaders as well as the 4As. And much appreciation, Jay. Thank you so much for your work. And the thing we got to remember too, I mean, this is a community of volunteers. Right? None of this would happen if people didn't step up like Jay and say, hey, I'm going to take that on. And so I do have a few business things to mention, trying to round up volunteers and things like that. But that's how this operates. I mean, it's not going to happen without your help. All right. Before we start officially, I've been doing some poems before each meeting. This time I've got a kind of a conceptual art piece, and it involves transforming a violin into tea. All right, and so I'll just play this. There's a classical music that goes with this, but I'll play this first. This will kind of set the tone for us. I think the time lapse was like a year-long time lapse of that growth, but fascinating to watch and definitely something you could drink. So just a couple quick announcements. Our fair coming up in August, we do need volunteers. All right. And so the QR code, if you want to access it, it'll take you to a Google form where you can kind of see what spots we need people to fill. And there are two different days. I think the 10th is the setup, and we definitely need helpers on that day. And then the fair on the 11th, we'll need many as well. So I'll leave this up for a second. I think you'll get announcements. You'll probably get an email blast here asking for volunteers as well. But definitely it's a great opportunity if you want to get involved and you want to learn. Definitely there's a lot of places for us. Did I put the date wrong? Oh, okay, thank you. So let me okay, let me on the 11 to 5. Okay, thank you for the correction. Cool. Thank you. All right. So another couple quick things. We are working as a group in iNaturalist. Is that how you call it, Joe? Project. Thank you. And if you're not familiar with iNaturalist, it's really a place where you all can participate as citizen scientists and contribute to the collective body of data that is out there. And so Joe took this on and kind of started us up, I believe, as a project. And so if you want to contribute, definitely, you can play a role in this and provide data for things that you're finding. And it doesn't take much. I'm converted. I initially very hesitant, but Andy thankfully convinced me that it's important, and I agree. So thank you. Let's do this. One other request for fair associated things. Our cultivation, I guess, display, one of the guys, Devin, he's asking for contributions. And if anybody wants to help out at the fair with cultivation things, if you have any contributions you could make in terms of blocks that are fruiting, I think definitely get in touch with Devin and he would appreciate the help. All right. And so before our speaker, I'm gonna turn this over to Andy Wilson, and he's gonna introduce our guest. So. And we mentioned that.
All right. Thank you, Greg. I'm Andrew Wilson. I'm the Associate Curator of Mycology here at Denver Botanic Gardens, and I might have talked in front of some of you before. And before I give our speaker an introduction, I wanted to point out that as the chair of the voucher collections project for the North American Mycological Association, the foray as an event, as registration opened today. So go to the North American Mycological Association's website and look up the foray for this year, which will be held in Randall, Washington, from October 31st through November 3rd. And that's a great event. People from all over the country come to collect mushrooms and fungi and learn about how to cultivate mushrooms and how to all their culinary wishes can be fulfilled at these meetings. But we also go there and we collect mushrooms from all over the region, learn about the diversity, which is something I'm really interested in, and as I'm sure you're interested in when you go on to forays as well. So check that out. And if you are interested, I suggest signing up as soon as possible. They opened up this afternoon, and I think they already got 150 registrants just today. So they'll probably sell out in the next couple of days. So that's just my little plug. So I'm happy to introduce Dr. Brian Perry. And is this your computer, Brian? Okay, so we'll plug yours in pretty soon here. Dr. Brian Perry began his foray into mycology at San Francisco State University back in the late 90s, mid-90s? Mid-90s, and I know this because he was my TA for mycology. We both began in the lab of Dr. Dennis Desjardin, which Brian will introduce you to pretty soon. And there he began his lifelong love affair of mycology and studying mushroom biodiversity. And as a master's student at San Francisco State University, became the foremost expert in the genus Mycena, probably in the world, but especially for California Mycenas. And if you're not familiar with the genus Mycena, they're very beautiful ephemeral mushrooms, and of course, going to be a central topic of today's talk on bioluminescent fungi, because the genus Mycena happens to be one of the centers of biodiversity when it comes to bioluminescent fungi. After working with Dennis, he went off to Harvard University to study under Dr. Donald Pfister, but in these Ascomycete cup fungi and the Pisces azaleas. He did a lot of cool things. Lose 20 pounds when traveling through China and the Himalayas because he can really only eat rice for a certain part of the time. And then after that, he bounced around a bit. He went back to San Francisco State to do a postdoc before getting a faculty position at the University of Hilo in Hawaii. And he was there for several years before ending up at California State University, East Bay, where he joins us from today. And I am happy to introduce my friend, Dr. Brian Perry, and he'll give a fascinating discussion about bioluminescent fungi, where they come from, and how they lighten our lives. So welcome, Brian. I'm going to go ahead and dim the lights for the audience.
Okay. Yep. So, all right. Thanks, Andy.
We'll stop sharing and you'll review.
All right, now they should be able to hear. Okay, sorry about that. And so in 2007, additional papers started to come out, and this was a publication that got the cover of Mycologia. And this was species Mycena lucentipes, and I'll show you more about this in just a few minutes. But before we get too far into thinking about bioluminescence, I want to tell you a little bit. Oh, my computer doesn't want to advance. Tell you a little bit about bioluminescence in general. And the first thing I want to distinguish between is really what is the difference between bioluminescence and fluorescence, right? So fluorescence has become really popular in the last couple of years. We're now using little UV flashlights when we go out mushroom hunting because many species of fungi will fluoresce, right? So what fluorescence is really the immediate re-emittance of absorbed light energy at a lower wavelength than at which it was absorbed, okay? That's very different from bioluminescence, which is the production and emission of light. And we can call these, you can measure these as photons by a living organism, okay? So what we're seeing in bioluminescence, as we'll learn tonight, it's chemical reactions that are occurring in the cytoplasm of the cells of the organisms that are luminescing, whereas fluorescence is really due to things like pigments in the surface tissues of various organisms that essentially will absorb light energy and then very quickly re-emit it. So it's really popular nowadays to go out and use a UV flashlight to look for fluorescent tissues on fungi. What you're seeing there in that photograph, that's the species Mycopan scabripes. On this side, this is no UV light. This is just normal natural daylight that this has been photographed in. In this photograph, I put a dark, basically light blocking sheet over the mushroom, and I shined a UV flashlight at it. And as you can see, this species is characterized by having this fluorescent yellow basal mycelium. And so it turns out this can be a really interesting and important taxonomic feature that's becoming more and more common when we do taxonomy of these species. And of course, most of us all know that many, many insects and other arthropods all tend to fluoresce. So if you've been into scorpions, you've gone out to the desert at night looking for these things, you can use a UV flashlight. You can trap insects by shining a UV light on a white sheet, and many insects will be attracted to that. So it's a really great way to collect insects. So that is fluorescence. We tonight are, of course, going to focus on bioluminescence. So we're going to be looking at a biochemical pathway that occurs in the cytoplasm of cells. And this is not unique to fungi by any means. Okay, so if we look at the tree of life, I'll show you another figure in just a minute, but kind of hard to see. Could we dim the lights so we can see the screen better? This is an anglerfish up here, made famous by such, fine movies as Finding Nemo. But what we see in an anglerfish is that it has a lure on the top of its head. It has this little projection. And inside of that lure, it harbors bacteria that produce bioluminescence, okay? This is a jellyfish here. Jellyfish have special organs that they produce bioluminescence in. These are glowworms. This is a cave in New Zealand. And what you're seeing here, these are essentially a sort of a fungus gnat type thing that produces these mucilaginous sheaths that they then illuminate. And they use that to attract prey, okay? These are dinoflagellates. I think that's San Diego in the background there. And what you're seeing here, these are dinoflagellates that are being tumbled in the surf. And this is something that happens periodically along the coast of the west coast of the US. If you go down to the Caribbean and places like that, it can be really common. And what's going on is as these dinoflagellates gets tumbled, they produce light. And they might be doing that for a number of different reasons. Of course, we have some insects that are bioluminescent. We have fireflies up here. As you all know, the male fireflies, actually both species sexes will luminesce during their nuptial flights. So they'll produce these bursts of light. Really amazing to see. This is a native millipede to the southern Sierra Nevada mountains in California, and it is a bioluminescent millipede. So you can go out at night in the southern Sierra, and if it's a moonless night, you can see the light of these millipedes. You can actually go out and find them and collect them quite easily. Pretty amazing. And then, of course, we have bacteria. This is a flask right here. You can see someone's hand, and this is just a colony of bacteria that is producing light. Okay. Now, if we look at the tree of life, what we see is that bioluminescence has evolved, we think, at least 40 times independently. Excuse me. So what that means is that over 40 times, the biochemical pathway that leads to the production of light has evolved in different groups of organisms. If we look at those biochemical pathways, what we see is that they are distinct, but they're also very similar in the sense of the way the chemical reaction occurs. I'll say more about that in just a few minutes. It occurs in a huge number of very distantly related organisms. And if you look at the tree there, that little arrow, there's fungi up there. So it occurs in over 10,000 species from 800 genera across 13 phyla. Okay, so the production of light from within the cytoplasm of the cells of living organisms is really widespread. Okay? We're simply going to focus. Oh, was there a question?
Yeah, I was wondering, it says luciferin type.
Mm-hmm.
Can you go over that a little? Is that how?
Yep, we are. So we're gonna look at that. It's gonna be it's a substrate enzyme reaction. I'll say more about that in just a sec. Yep. All right. So when we look at these, of course, one of the biggest questions that we always are asked, and I'm sure you will have wonderful questions tonight, is why are they doing it? And I'm going to tell you what our hypotheses are and why we think they're bioluminescing. But when we look across the tree of life, we see that organisms are bioluminescing for a lot of different reasons. When we look at fireflies, we know they're doing it to attract mates. Okay, if we think of something like an anglerfish, they're doing it to attract prey. If we look at dinoflagellates, they're probably using a startle response to prevent things from eating them. Jellyfish, they're using it to also attract other individuals as mates, but also perhaps to deter things from eating them. These glowworms are simply using it to attract prey as well. Okay, so we see it used and it's evolved a number of times independently for many, many different reasons. And when it gets to fungi, well, I won't spoil it now, I'll wait till the end, but we have some ideas, but we don't know for certain, right? But there's lots of fun things we can hypothesize about. So what do we know about fungal bioluminescence? Well, we can go back to the sort of earliest reports in the literature, and that goes all the way back to Aristotle. So Aristotle talks about glowing mushrooms. Oh, no, excuse me, that's Pliny. Aristotle talks about glowing wood. So he was one of the first folks to distinguish between wood that was glowing because it had been burning, so embers or coals, versus glowing because there was something growing on it and producing that light. Okay, so Aristotle references this in his writings. Pliny the Elder, not that one, but that Pliny the Elder, he talks about glowing mushrooms in the tops of oak trees in the Gallic provinces. Okay, so here he's talking about actual bioluminescent mushrooms occurring in Europe. A little bit more recently, Fernandez in 1526 talks about Spanish soldiers in the West Indies using little pieces of bioluminescent bark, so little glowing chunks of wood, and putting that on their helmets or someplace on themselves so they can see one another at night. And there's similar reports from Ramsbottom about soldiers in the trenches of both World War I and World War II, putting pieces of glowing bark or glowing mushrooms on their helmets so they could see one another in the trenches, right? Because of course you never want to use a flashlight in the trenches because of snipers, the whole thing about lighting a match to light cigarettes, you can do it very quickly. Bioluminescence can't be seen from that far away. So it was actually a safe way to illuminate and identify one another in the trenches without alerting, the opposing troops. Olaus Magnus talks about in Scandinavia using bark or glowing mushrooms to go into barns at night or to find your way in the woods. If you think about it, barns are full of hay, highly combustible. If you go into a barn with a lantern, very easy to catch that barn on fire. So what they would do in parts of Scandinavia is that they would use glowing mushrooms to go in and illuminate the barn. Pretty neat, okay? And then if you look on a broader scale around the world, there's all sorts of wonderful reports of both ritualistic and practical uses of both bioluminescent mushrooms, glowing pieces of wood, or bioluminescent mycelium from various cultures around the world. A little bit more closer to home, we have the use of bioluminescent wood in America's first submarine. Okay, so the Turtle submarine was built in 1776, so during the American Revolution. It's the first submersible built by Americans, and it was designed to go out into harbors where the Royal Navy British ships were, and you can kind of see from the diagram there, it has a little drill. So this thing would go out into the harbors at night, it would go up to these ships, it was supposed to drill a hole in the bottom of the ship, insert explosives, and then paddle away very, very quickly before the ship exploded. Okay, now this is a sealed contraption, right? So here's the operator, here he's got some foot pedals and cranks and all these sorts of things. He's got to find his way in the dark to these ships, drill a hole in it, insert some dynamite, and then, basically light it and paddle away. Well, in order to see the controls, he needs light inside of that contraption. Now, if he used a candle or a lantern, what would happen? He'd suffocate, right? So that candle is going to essentially consume all of the oxygen that would be present inside that chamber with him. So instead, they used glowing bioluminescent wood. We call this foxfire. Okay, so foxfire is the wood of various different tree species that is infected with the mycelium, usually of species of Armillaria. In the southeastern United States, we call that foxfire. We can find it out here as well in the West. Okay, so they use that to illuminate the inside of the turtle submarine. Submarine was never successful. It never sunk any ships. Eventually, it was sunk itself, or the ship carrying it was sunk by the British Navy, and so it's on the bottom of some bay someplace on the East Coast. But there's a replica of it in a museum that you can go check out if you want. All right, so what do we really know about fungi? And how many species of fungi are bioluminescent, and how are they doing it? Well, this number keeps increasing. So I had to go in tonight and change my slide to over 130 species. We just submitted a paper for review that is a really nice overview of everything that we know about fungal bioluminescence. And to date, we have 132 species in that paper that we are listing. Since that paper was submitted for review, there's already been a handful of new species reported as being bioluminescent. So this number is always increasing because people are out there now looking for it. Okay, so at least 130 species or 132 species. Now, what do we know about the actual biochemical pathway that produces light? So what we see is that it is a luciferin and luciferase reaction. Okay, so luciferin is a substrate molecule. Luciferase is an enzyme. And in the presence of molecular oxygen, as well as NADPH, we know that there is a reaction that occurs. It produces oxyluciferin, water, and light. And this is a really basic formula that we've known about bioluminescence for a number of years, going probably as far back to maybe back to the 60s. We've known this, maybe into the 50s. And so if we look at fireflies, or we look at many of the other bioluminescent organisms out there, or we look at fungi, we know that it's a similar type of reaction. It's always a substrate molecule being acted upon by an enzyme, and the end product is going to be an oxyluciferin and light. What's different, of course, is that the substrate molecule that the system is utilizing, as well as the enzymes that drive the reaction, are different in all these different lineages that have evolved bioluminescence. So the term luciferin and luciferase are very generic terms. They simply refer to a substrate molecule and an enzyme that catalyzes the bioluminescence reaction. Okay, so when I say here, it said, so we know that this is really, really common. So if we look at the way this occurs in fireflies, it's very similar to what we see in fungi, but there's different types of luciferin, and we didn't know it back when this slide was made, but now we actually know what the enzyme is in fungi. It's obviously a very, very different enzyme than what we see occurring in fireflies and other bioluminescent species. And this is an important one here. This kind of starts to hint at something that we'll talk about, and that's how many times did fungal bioluminescence evolve? Well, all fungal species that are bioluminescent emit light that is greenish yellow in color, as you can see in the photograph there of Mycena lux coli. And they're all within the range of 520 to 530 nanometers. Okay, if you go back and look at really old reports, they'll talk about a species having blue luminescence coming out of them, and we've never been able to verify any of those reports. There's a lot of chemiluminescence that occurs in a number of organisms, very, very different from bioluminescence as we are referring to it here. Okay, so here's a nice chemical diagram for you. I'm not going to go through this in gory detail, but what I do want to point out about this, and what makes fungal bioluminescence very unique and very different from bioluminescence in other organisms, is that it happens all the time. Okay, so if you think about fireflies, when do they produce luminescence all the time? They just give little bursts of light. Okay, if we think of dinoflagellates, like, so if you've ever been out to a bay and paddled through, or you've ever swum and, in a bay where there's dinoflagellates, they produce a flash of light and then it gets extinguished, and maybe they'll flash again, but they have to essentially renew the ability to do it. It's not constant. Fungi are the only organisms that have the ability to bioluminesce 24 hours a day, 7 days a week. And the reason for this is that the biochemical pathway that produces bioluminescence is cyclic, right? And so it's all based on this. This is caffeic acid. Caffeic acid is a really, really common plant metabolite. So most plants produce it. Different plants produce, differing amounts of it. That is the precursor molecule. It essentially gets transformed by histidine synthetase. So here's one enzyme. So what we're going to see is we're going to get this cyclic pathway, which is catalyzed by a series of different enzymes. And all of these enzymes, or at least most of them, depending on the species, are going to be encoded for in the genomes, I should say, of these fungi. So here we have one enzyme that's going to convert caffeic acid into histidine. That is fungal luciferin, right? That's the substrate molecule that the enzyme is going to operate on. So that's going to get modified by another enzyme here. It's going to become 3-hydroxyhistidine, and this is sort of the activated form of luciferin. LUZ here is the luciferase. This is the enzyme that is catalyzing that reaction. So in the presence of molecular oxygen, we're going to see that luciferin gets elevated to this high energy intermediate, and then it very quickly decomposes back to a lower energy state. And when it does that, it produces light, okay? So it gives off light as the excess energy. So rather than releasing that energy purely as heat, as most reactions would do, it's releasing that excess energy as light. It produces carbon dioxide here. And then this is the oxyluciferin. This is the oxidized form of luciferin. It gets converted by yet another enzyme back into caffeic acid. So it's a cyclic pathway. So as long as the fungus has a supply or a starting supply of caffeic acid in the presence of molecular oxygen, it has the ability to go through this biochemical pathway and produce light, which is unlike bioluminescence in any other organisms we've studied so far. That's what makes it really unique. It's also what has allowed folks to take this pathway and insert it into other organisms. So how many people own a firefly petunia? Anyone in the audience? No one? So you can order bioluminescent plants now. Okay, so the folks that published this paper back in 2018, several of them have founded a company with some collaborators. They've taken this pathway, really the genes responsible for this pathway, and inserted it into different organisms. They put it into a tobacco plant first, and now they've put it into a petunia. And you can order these petunias online. They come, you can have them sitting on your dining room table. And when you turn off all the lights, the whole plant glows, right? So they're using this, they're able to take this whole pathway and insert it into different organisms. So pretty amazing. But they're only able to do this because of the cyclic nature of the pathway. Okay. So what else do we know about them? So who are these bioluminescent fungi? Well, all known bioluminescent fungi are mushroom-forming members of the Basidiomycota, and they're all within the class Agaricomycotina. So the Agaricales is a name you might know. They're all members of the order Agaricales. They're also all white-spored species. And back in the old days, we used to put all of them in this family, the Tricholemataceae. Nowadays, as you'll see in a minute, they're sort of spread out into a bunch of different families, but they're all white-spored, mushroom-forming species. All of them are saprotrophs. The vast majority of them break down dead organic matter in the form of wood. So many of them are wood rotters, where they're breaking down cellulose and lignin. And that's an important thing to keep in mind for later in the talk. And then there's one species that's a parasite on coffee. I'll show you some photos of that one. And they occur in not 4, we used to think it was 4, but we just recently discovered a 5th lineage of bioluminescent fungi. We published this earlier in the year. So we have 5 distinct evolutionary lineages within the order Agaricales in which bioluminescence occurs. And so this horribly confusing diagram, all this is if you're used to normal evolutionary trees, which might just be these long, tall things, I've just taken one of those and I've just folded it back on itself to make it more difficult to read. Okay, so just because it's the one way you can get it to fit on a single page. So these are becoming more and more common as we have, these gigantic evolutionary trees that have thousands and thousands of species. This one is a tree of, I think, about 3,500 species. This is all the order Agaricales. So here's the Boletales, so the boletes right here, that's the outgroup. And then all these other things you're seeing here, these are all different families of Agaricales, okay? So this is from this is the review paper that we have that's in review at the moment. And if we go in and look at the 5 bioluminescent lineages, I've highlighted those in bioluminescent green there on the tree. I can zoom in on that to make it a little bit easier to see. And so what we see here is that we have 5 lineages. There's the Omphalotus lineage, the Armillaria lineage, the Lucentipes lineage, Mycenoid, so Mycena and relatives. And then this is this one that we just discovered earlier this year, the Eocyphella lineage. Okay, so 5 lineages all of which contain bioluminescent species. Okay, we'll come back to this later in the talk, but notice that there's no green anyplace else in the tree. It's all right in this area here. And this suggests that perhaps there was a single origin of bioluminescence in the fungi. Question? Yes and no. So what we see is that in some groups, there are non-bioluminescent members that have lost one or more of the genes responsible for the pathway. But when we go and look at other groups, other families, there are typically none or only a few of the required genes. So the whole pathway has never been present in those. I'll come back to this at the end of the talk. We have other information that informs this. But what it's suggesting right now is that there was a single origin of bioluminescence in the fungi, but as I'll talk about later, probably many, many losses of it, okay? And so now what we can do is we can walk through these lineages and look at some pretty pictures of glowing mushrooms. All right, so the Omphaliotis lineage, we'll start here. 18 species in the Omphaliotis lineage that are known to be bioluminescent. And those, of course, include things like Omphaliotis olivascens, which you folks, do you have this out here? I don't think you do. I don't think it occurs this far east. This is something that was described from California, pretty common in California, also occurs up into Oregon. I wanted this to be the California state mushroom, but they chose the golden chanterelle instead. Boo. But Omphaliotis olivascens, it's the western counterpart to the eastern jack-o'-lantern mushroom, which I'll show you a slide of it in just a moment. Really common in the Pacific Northwest. It's poisonous to mammals, so we cannot eat it. No other, animals with no other mammals are going to eat it. Insects will certainly eat it. And in this case, you'll notice in a lot of these slides, I'm going to tell you whether or not the mushrooms and the mycelium glow, or is it just the mycelium that glows? So one of the other interesting things we see about bioluminescence is you can have bioluminescent mycelium, or you can have bioluminescent mycelium and mushrooms. But we don't get situations where there's just bioluminescent mushrooms and non-bioluminescent mycelium. And that just doesn't occur. So Western jack-o'-lantern mushroom, when I give this talk any place in California or the Pacific Northwest, I always tell folks, this is your best bet to see a bioluminescent mushroom locally. These are big, they're easy to find. If they're in good shape, meaning they're nice and fresh and not starting to rot and you bring them home, they can be incredibly bright. I had some in my, that I collected last fall, brought it home, put it in the middle of the day. I just went into my bathroom and closed the door, and within 5 seconds, I could see this thing glowing on the counter. So incredibly bright. Other times I've collected it though, it's been a little bit older, a little bit dried out, and it barely glows at all. And if you're artistic, you can get nice slides where you get both the glowing portion and sort of what it looks like in the light as well. This is a photo from Trent Pierce, who is actually lives up in Boulder now. He's a local person, so maybe you can get him involved with CMS. Used to be in the Bay Area. And then also in this Omphaliotis lineage, we get things like Neonothopanus nambii. This is something that's common in both the Paleotropics and the Neotropics. We don't know if the mycelium is luminescent because we've never been able to get this thing in culture. When we go and do field work in the tropics, it's really hard to bring living material back into the US. You have to have a whole lot of permits to do so. So when we collect these mushrooms, we photograph them and then we dry them, right? So by the time we bring them back, they're dead and crispy and no longer viable. So we've never had a chance to get this one in culture to see if its mycelium is luminescent. It has to be because of all the other ones we know about. If you've got glowing mushrooms, you always have glowing mycelium, so it should be for certain. We don't know if it's poisonous. We've never tried to eat this one. Given the fact that it's closely related to Omphaliotis, it probably is. The locals where we've collected this in Micronesia, they do not eat it, but their common name for this is ghost ears in Micronesia. And the reason for that is because it grows on the sides of logs and trees and is kind of ear-shaped. When you go into the forest at night, it looks like there's glowing ears on the sides of logs and sides of trees. So the common name for it in many of these spots is ghost ears. Okay, and this is what it looks like when you turn out the lights. So a really intense, beautiful mushroom. This is probably one of the more brighter species that I've ever photographed. And also in this group, there's another Neonothopanus species that's important for us. And this is Neonothopanus gardneri. And this is one that is from Brazil. And this is the mushroom that has really made it possible for us to understand the biochemical pathway that leads to bioluminescence. And that's because it's big and you can collect a lot of it. So this thing is really cool. Almost looks like a chanterelle or something. This is growing at the base of a palm. We don't know if it's parasitic on these palms. We think it might be a weak parasite on these palms. It always grows in these big clusters at the bases. This was first collected in 1840 in Brazil, and it was described as Agaricus gardneri. It was never known to be bioluminescent until our collaborators, Marina Capillari in particular, and Cassia Stevani, went and started collecting this thing in Brazil, brought it back to the laboratory, and realized that it was intensely bioluminescent. So we were able to get our hands on it, figure out where it actually belonged evolutionarily, moved it into the genus Neonothopanus. And it's really important because, as I said, you can collect a lot of it. So many of the bioluminescent species I'm going to show you tonight are incredibly small, tiny mushrooms, and you can usually only find a handful of them. This one, you can collect gobs of it when you go out at the right time of year. It's also very easy to get it growing in culture. So you can grow huge amounts of mycelium, which you can use in the laboratory. And in this slide, you can see this is the palm that it grows on. This is a sterilized bag of those palm fronds that's been inoculated with the mycelium of the species. You can see how intensely it glows. And so this mushroom, because we could collect a lot of it, and because it was very easy to grow large amounts of mycelium in the lab, this mushroom was responsible for our biochemist colleagues to go in and do the chemical reaction studies that allowed us to elucidate the luciferins and luciferase, which led to the discovery of that whole genetic pathway leading to bioluminescence. So without this mushroom, we probably would have taken a lot longer to get there. All right, now onto one you're probably much more familiar with, and that's Armillaria, right? So the honey mushrooms. And so Armillaria mellia, that's a name that we have used for many, many years in the Pacific Northwest for what is really a complex of at least a dozen species. I don't know how many species of Armillaria you have here in Colorado, but I'm guessing plenty. And they're, these mushrooms they can be pretty variable, but they almost always grow in these really massive clusters on logs. Some of them are saprotrophs, but the majority of these species are root pathogens. So you've all heard of the humongous fungus. You've probably seen it on the sides of U-Haul vans and things like that. That's a species of Armillaria, right? So in Oregon and in Michigan, they have these huge populations. They've been used genetic tools to confirm what they think is that, these massive populations of this mushroom appear to all be one genetic individual that's spread over, thousands of hectares. So they're the humongous fungus species. And people eat these, right? So these are known as honey mushrooms. All of them are edible. What people tend to do is they'll collect these really large clusters of them, then they eat the stipes. I've never eaten this thing. Seems like too much work to me, but they can be really common. And this is an important bioluminescent species because this is what produces foxfire. So in Armillaria, the mycelium and then the rhizomorphs, and rhizomorphs are these sort of thick cord-like masses of mycelium that become all melanized. You can see some here, see these dark things? Those are just masses of mycelium that become really hard and almost carbonaceous. And they do this so that they can persist in the environment. When those are young and they're just being formed, which you can see in this Petri plate here, they are bioluminescent. And then the mycelium itself is bioluminescent. So I was telling folks at dinner that this is probably one of the easier bioluminescent mushrooms to find here in Colorado. If you wanted to do a nighttime foray when you went out looking for bioluminescent species, going and finding logs that are being broken down by Armillaria species and start pulling those logs apart. And of course, doing this on a moonless light, turn off your headlamp, your flashlights, let your eyes adjust, you'll typically be able to find chunks of glowing wood, right? And the term for that is foxfire. So for a number of years, we thought that it was only the mycelium and the rhizomorphs that were bioluminescent because we had never seen the mushrooms of Armillaria bioluminescing until Taylor Lockwood went down to New Zealand and photographed this. This is Armillaria limonea, and this is what it looks like in the dark. So the caps, especially the margin of the pileus, are really intensely bioluminescent. No one had ever recorded this. So here's our first example of mushrooms in Armillaria that are actually luminescent. Not too long after that, our collaborators in Brazil sent us these photos, and this is another Armillaria species from Brazil. It's very small, it's probably only an inch tall, but again, when you turn off the lights, the cap glows as well. And so we're in the process of describing this as a new species right now. So it does turn out that when you get into perhaps the Southern Hemisphere, the mushrooms of Armillaria are luminescing, but we've never seen this in the northern temperate species that we have here. Don't know why. Could be that the substrates they're growing on aren't producing enough caffeic acid. So maybe it's something where we could play around with them and spray them with caffeic acid to see if they'll glow. All right, and then the Lucentipes lineage. So this is an interesting group. This is two species, Mycena geronima viridolucens, Mycena lucentipes. These are two species that were published right when I started my postdoctoral work. First reported bioluminescent species of Geronima, not a widespread genus in the Northern Hemisphere, much more common in the tropics in the Southern Hemisphere. So you can absolutely find this if you go down to Brazil, in the tropics and places like that. We'd never seen any bioluminescent Geronima species. So this was the first report of that. Turns out it's not a Geronima. Okay, so Dennis Desjardin was the one that sort of did the morphology studies on this, thought it was a Geronima. I came along as part of my postdoc and sequenced the DNA from it, did some analyses, and was able to tell him that it's not a Geronima. It turns out it's a new genus, right? So this thing falls out completely on its own, doesn't form, good relationships with any other species that we've ever sequenced to date. So this seems to be a genus of one species, only known from the Atlantic rainforest down in Brazil so far. And in this case, it's really just the lamellae of the mushrooms that are luminescent, but it's bright enough that you can actually see it through the cap. It's pretty cool. And then there's this one. This is Mycena lucentipes. These two were published at the same time. Just like Geronima, turns out this one is not a Mycena at all. This actually goes in a totally different part of the evolutionary tree. But this species, it's actually really quite common when you get down into the Atlantic rainforest in Brazil. When you get down there, if you go out at night, especially in Pitar, which is one of the national parks down there, you can find gobs of this mushroom. So go out at night when there's no moon, turn off your headlamp, your flashlight, and you will find this mushroom in abundance if you hit it just right climactically. Dennis described it as a species of Mycena, but I came along again and sequenced the DNA, did a bunch of analyses, and it turns out this is also a completely separate genus. We now know better where it goes. It's actually, well, it was also the cover of Mycology. So the same week this journal came out, and Dennis was super proud of having the cover, I got to walk in and tell him that the species he described as a Mycena wasn't a Mycena, which you can imagine didn't make him terribly happy with his postdoc. But this thing's actually a species of Mycopan. So based on all the analyses that I've done, if you know the genus Mycopan, it's kind of looks just like the Mycena, but its molecules tell us it's in a very different part of the phylogenetic tree of fungi. This thing is actually a Mycopan species. So we're in the process right now of transferring this into the genus Mycopan. And of course, this is the species made famous as the first mushroom on a US postal stamp. This was Taylor Lockwood's photograph of Mycena lucentipes. One of Taylor Lockwood's lifelong dreams was to get one of his mushroom photographs on a US postal stamp. So when he heard that they were doing a series on bioluminescent life, he submitted his photo and he got it on there. And so Taylor was absolutely delighted. We were delighted that we actually got a bioluminescent species of mushroom on a US postal stamp. All right, and then finally into the big group. So as Andy talked about before, some water here. I've been working on the genus Mycena in what I refer to as the mycenoid fungi for years. Ever since I was actually an undergrad is when I first started working on these. So they've been sort of my first fungal love. And it just happens that the mycenoid fungi contain the largest concentration of bioluminescent species. We don't know why. They're an incredibly diverse group, so it probably just has to do with that. There's 96 species so far of Mycena that we have documented. And I'm going to tell you that there's probably hundreds, if not thousands, of additional species of Mycena that are also bioluminescent. It's an incredibly diverse group of mushrooms worldwide and pretty poorly documented from around the globe. So every day, people that are sort of into this sort of thing are going out. Many of our northern temperate species of Mycena are actually luminescent, but not at levels that human eyes can detect. So you can do things like take a digital camera and set it up to do a 15-minute exposure, and it'll resolve some glowing mushrooms, but never at levels that our eyes will ever see. We also have really fancy machines called luminometers, and those luminometers actually measure light output. So I have one of these in my lab, so I can take one of these mushrooms, put it in a little glass jar, put it in the machine, and it will read any light that mushroom is putting out. Okay, so what we're doing is we're trying to go through and document more and more of these species that are actually luminescent. This is a species that you folks definitely have here in Colorado, and it's also one that you can order. You can get the mycelium of this from a culture supply house. You can grow this thing at home. This is Pinellus stypticus. Who's seen this mushroom? A couple of you. So Andy, I see your hand out there. A few of you. It's not very big, so the largest you'll ever see it is maybe, I don't know, an inch in diameter, but it can be really common during certain types of years. It likes to grow on hardwoods, so go into habitats where you're going to find hardwoods. It's really common worldwide, primarily in the Northern Hemisphere. It's inedible, it's really bitter, it's styptic, so it's really astringent if you put it in your mouth. And what's really neat about it is that the mushrooms of the mycelium glow in Eastern North American populations, but they don't glow in European populations. Right? So here we have probably a situation where it's lost one or more of the genes in these European populations that are genetically isolated. For the longest time, we also assumed that Western North American populations also were non-luminescent until really recently. So Nathan Warner, who's one of the California Fundus Project collectors, he went out this past fall and winter, collected Pinellus stypticus, brought it home and did this incredibly long timed exposure with his digital camera. And sure enough, at least some of our Western North American populations are bioluminescent. So if you can find this mushroom here in Colorado, chances are it will be bioluminescent. So that would be another one that you could look for. And then within the mycenoid fungi, I could talk for hours and hours about all of these. I'm just going to kind of go through these just to show you as many pictures as I can. Most of these are going to be species that we find in the tropics. Okay, so species of Mycena that we have in the Northern Hemisphere, there are a number of bioluminescent species, but most of them luminesce at really low intensities. That's really difficult for our human eyes to detect. When you get down into the tropical zone, that's very, very different. You can go out into the jungle at night and find lots of glowing mushrooms, as well as the wood and the leaves that these fungi are growing upon. Okay, so this is Mycena chlorophyll, really common in the tropics, probably one of the most commonly observed and photographed bioluminescent species in both the paleo and the neotropics. Really common. Come on, computer, you can do it. Mycena lux coli. This one is common in the Asian tropics. And I love this one simply because there's been some really nice work showing that the mushroom is eaten by nocturnal spiders. Right? So these glow-in-the-dark mushrooms have these spiders that come down out of the trees at night, and they will actually consume portions of the mushroom, or perhaps they're consuming really small insects that are also feeding on the mushroom. So pretty cool that it has this relationship with some nocturnal spiders. Mushroom also comes in both green and red. So you can take your pick, whichever you like most. Both color variations are bioluminescent. We do get one parasitic species. It's really a pathogen. This is Mycena citricolor, and this causes American leaf spot on coffee. So it's a really virulent pathogen, especially in South America and in places like Hawaii and other coffee growing regions of the world. They do a lot to prevent this fungus from getting there. And it's not the mushrooms that are luminescent in this species, it's their mycelium, but they also produce these really bizarre little asexual structures called stilboids that are really just little hyphal things that are designed to disperse this fungus so it can spread throughout these coffee plantations. So this is what they look like, this nice little drawing. They're just these little hyphal strands that come up, and then there's this little head on top that disarticulates. If you look at that thing under a microscope, it's got all these little sticky hairs all over the surface. And so when rain comes or wind comes, these things break off and it helps to spread that fungus. So because it's a pathogen, it helps it to go on to infect other parts of the plant and other individual plants. And here's just to show you, this is a 1 millimeter scale bar. So here's the stilboid, this is maybe over 1 millimeter tall, like 1.5 mils. And then here's the actual mushroom of that same species. So it's bizarre. Mushrooms aren't luminescent, but the stilboids and the mycelium are. So if you were a coffee grower and you went out into your coffee plantation at night and all the leaves were glowing, you'd know you were in trouble because you have the fungus that causes American leaf spot of coffee. And then there's this one. This is probably the most commonly photographed bioluminescent fungus in the world. This is Phylloboletus manipularis, really common in the Asian tropics and throughout the Pacific on decaying wood. Andy and I did, it actually it occurs around the world. Andy and I did field work in Borneo. And we collected a log that was just covered with these. And I made a big collection of it. I photographed it, which you're looking at here. And then I took some of the extra mushrooms and I just put them on the nightstand between the beds in the guest house where Andy and I were staying. And this thing glowed all night. So it was just like having this mushroom, glowing nightlight. And it, continue to glow until the mushrooms dried out. So these things will bioluminesce for hours and hours and hours until those mushrooms lose enough moisture and they start to rot. Really, really common. It's described as Phyloboletus. It's actually a Mycena species. It's been called both. So Mycena manipularis is actually the proper name for this species. The other thing that's kind of cool about it is it appears to be, it might be invasive, right? So this thing is really common throughout the Asian tropics, throughout both the neo and the paleotropics. This is one from Micronesia. So we turn out the lights, it looks like that. This thing was recently picked up in Hawaii. So it showed up on Kauai about 10 years ago, and now it's becoming more and more common on Kauai, and it's also showing up on Oahu. So it's made its way to Hawaii. So who knows, maybe even we'll start getting it in Southern California and some more warmer, humid climates. And then lots more. So if you've ever seen Roridomyces rorida, species that we have here in the West, that's this one up here. It's a Mycena that has this really thick mucilaginous layer on the stipe. It has been described as Roridomyces. Again, it's the molecules tell us it's actually a species of Mycena. When you get down into the tropics, many of them have the base of the stipe is bioluminescent. What this is, it's the mycelium that's luminescing, and that's where the mycelium is attaching that little mushroom to the stick. And so we're always curious whether or not our North American species would luminesce. And so a couple of years ago at Soma Camp, I'm sure you all know Alan Rockefeller, this little Mycena rorida came in, it was growing on an acorn. And so Alan had brought it up and showed it to me, and I asked him, I said, can you go photograph that? And let's see. So Alan went into a dark bathroom at the camp there, set his camera to open his lens all the way, set the sensitivity to its maximum, and did a 220-second exposure. And then he turned on the bathroom light. So we got both the bioluminescent mycelium of this species, as well as, you can see what it looks like in the light. And so here we do absolutely have bioluminescent species in North America from this group, but Alan was not able to detect this bioluminescence with the naked eye at all. So not glowing at levels here that we can see with our own eyes, but our cameras can see it, luminometers can see it. All right, and then this is a fun one. This was, again, Andy and I did some work in Malaysian Borneo. This is a species that I described from down there, growing on the sides of trees. Really pretty, Mycena sylvolucens. We get Mycena lux eterna. So Dr. Desjardin really loves to use sort of interesting names for these. And so some of these names are actually taken from like Mozart's Requiem and things like that. So the eternal light is lux eterna. So again, this is a species known only from Brazil. Incredibly bright luminescent stipes. No, they're really beautiful species to see down there. And then we'll end with this one. So I've been doing field work in Vanuatu in the Pacific, South Pacific for a number of years. And as you can imagine, there's a lot of bioluminescent species down there. This one is my favorite. This is a species of Mycena that is undescribed, so it's new to science. So I'll be putting a name on this at some point. And when you see it in the dark, it has these beautifully bioluminescent stipes. It's pretty nice. All right, and then we'll just end with the last one. This is the Eocyphella lineage. My computer keeps freezing on me here. I'm getting the spinning wheel of death. There it goes, sorry. This is Eocyphella lucior oleata. And this thing was discovered in Brazil by our collaborators. And this is a tiny little cyphaloid fungus. So cyphaloid fungi are these highly reduced forms. Many of them are less than 1 millimeter in diameter. This thing, I want you to look at the scale bar here. That's 500 microns, right? So each one of these is about 200 to 300 microns wide. So incredibly small. There are just these little teeny cups that are hanging upside down on the underside of branches and on the trunks of trees. And it's just the margin of the cup right here where it luminesces. And there's always water droplets right on the margin of the cup, which we think act to magnify and make that luminescence even more intense. And so this was the 5th lineage that we just recently discovered. So this was a really interesting find, but it goes to show that if people look hard, we're definitely going to find more and more species of bioluminescent fungi out there. All right. And so now the question you've all been waiting for, why do they do this? And folks have hypothesized about this for a long, long time. So as far back as the 60s, people were hypothesizing that perhaps these mushrooms were glowing because they were trying to attract animals to aid in nocturnal spore dispersal, which sounds pretty plausible. right? And that makes a lot of sense, except that for many of these species, the mushrooms don't glow, only the mycelium does, right? But there's been some really nice work done. In 1981, a researcher by the name of Savinsky in Florida went out and he took Dictyopanus pusillus, which is closer to Pinellus dipticus, and he grew it on leaves and other substrate. And so he got the mycelium covering that so that it was luminescing. And he put that in jars and he painted the outside of the jars with sticky stuff. And then he took jars that had the same substrate material but wasn't glowing, and he put that in jars and painted it with sticky stuff, and he put these out. And he did a whole bunch of replicates out in the woods where he was in Florida. Came back the next day and was able to show that the jars that contain the luminescent mycelium were covered in way more insects than were the jars that contain no luminescent mycelium. So he used that as evidence to suggest and to, hypothesize that perhaps these fungi are glowing to attract these animals. Those animals might act as spore dispersers, or they may be attracted to the glowing mushrooms and then they'll come eat the other fungivorous insects that otherwise would eat those mushrooms before they had a chance to produce their spores. Okay, which sounds great, which is probably, it's a viable hypothesis of why these things might bioluminesce. They attract predators of insects, as I mentioned before. So again, attracting predators will come eat the fungivorous insects that would otherwise eat the mushrooms or perhaps even the mycelium. It's been speculated that perhaps it's a warning coloration, so an aposematic coloration like we see in many insects, but that doesn't really hold up. Okay, so it doesn't really matter if you're a glowing mushroom or not, some insect is going to eat you. Okay, so whether or not they're glowing doesn't seem to prevent anything from eating them. So that one doesn't hold up. And then even really toxic mushrooms, they're typically toxic to things like mammals or vertebrates. Insects usually have no issues eating mushrooms, even ones that are deadly poisonous to us, of course. Okay, so it doesn't really work for the luminescent ones, and most of those are not toxic anyways. And then finally, we get to this one. And this is this idea that luminescence is an energy-releasing byproduct of an otherwise important metabolic pathway. And I told you earlier that most of these species break down dead organic wood as a food resource, or they're just saprotrophs breaking down cellulose and things like that. Well, several compounds in the biochemical pathway, turns out the one that produces luminescence, are really powerful antioxidants. And so part of breaking down lignin, which is a major component of wood, that's what gives wood its structural soundness, that lignin, when it gets broken down, produces reactive oxygen species, which can be really damaging to other molecules within the cytoplasm of cells in which that is occurring. So what may be going on is that by producing luminescence, it's actually allowing these fungi to have an antioxidant pathway in their cytoplasm that helps them deal with the molecules being generated from the food that they are eating. Okay, which makes a lot of sense when you think about it. So if we look at all these mushrooms that are bioluminescent, almost all of them break down wood. So this idea that this is simply just a byproduct of a metabolic pathway that allows them to be successful at breaking down wood, and they're simply releasing that energy as light rather than heat, it makes a lot of sense. Okay, and so that's sort of what we're thinking is probably what's going on. So we know that all saprotrophic fungi require antioxidants for efficient litter and wood decomposition. And we think that this bioluminescent pathway is probably playing a role in that. However, what I always say here is that it doesn't mean that that production of light hasn't been co-opted for some other evolutionary purpose. Okay, so these studies showing that insects are attracted to light, those have been redone. And they've been shown over and over again that insects are truly attracted to glowing mushrooms. So it could be that it's allowing them to break down wood, but evolutionarily it can also be selected for because it's helping them to disperse their spores or to attract, predaceous insects that will come and eat insects that are otherwise eating those mushrooms and mycelium, which is pretty cool. All right, so to wrap this part up, so what do we know? Is this a common phenomenon in fungi? And it turns out it's not. It's actually incredibly rare, right? So there's about 1.5 million species of fungi, and that's sort of our lowest estimate. About 10,000 to 12,000 are known to be mushroom-forming species. Luminescent species of mushrooms represent less than 1% of the agaricales and less than 0.005% of all fungal species. So bioluminescence in fungi is actually incredibly rare when we look at the entire kingdom fungi. Okay, it occurs in 5 distinct evolutionary lineages. They all luminesce in the same range of wavelengths. And of course, we all know now, we've talked about this before, but it's a similar pathway. So as far as we can tell, it's the same pathway occurring in all of these species. So when I got into this work as a postdoc, my job was to try and figure out, did bioluminescence evolve a single time in kingdom fungi, or did it evolve multiple times independently, like we see across the tree of life? And I, of course, was doing phylogenetic systematics, so I was generating, basically big evolutionary trees like this based on DNA sequence data. And back in those days, remember, this was way back in like 2009. So in those days, what we would do is we would build these big data sets, these big trees, and then we would go in and we'd use these really sort of bad algorithms to generate statistical inferences about what sort of character state might be present at different branches in the tree. And so what you're looking at here, this is just a tree. Here's the mice. This is basically all 4 of the lineages we knew at the time. And I'm going in and I'm asking, what's the probability that the ancestor to each of those lineages was luminescent? Okay, so Mycena up here, 90%. This one, the Centipede clade, 93%. This one for Amphlotaceae, 55, and then 86. Okay. Now, as you might imagine, these are sort of what we call scientific wild-ass guesses for doing this kind of work, and they're completely biased by the number of bioluminescent species you have in your sample here. Okay, so this was just sort of our best guess at the time, and really easy to bias these types of analyses. So they're unsatisfying, but at that point in time, it was what we had available. And if you go and you look at the base of the Agaricales, I was able to share that 65% probability that the ancestor to all Agaricales was bioluminescent, which is unequivocal. It doesn't tell us anything, right? So pretty unsatisfying. So fortunately, we were working with the biochemists, and Anderson Oliveira was doing his postdoc at the time. He came up to San Francisco when I was a postdoc there as well. And what he did is he went and he took mushrooms, and doing both what's called a hot extract method and a cold extract method, he extracted the luciferins and the luciferases. And so he could take a mushroom like this. This is our Neonothopanus gardneri. He could extract the enzyme and the substrate molecules, and then in a very controlled environment, he could mix those 2 things together and they would produce light. So you could recreate the bioluminescent chemical reaction in a test tube in a highly controlled environment. And so what we were able to do was Anderson went and he extracted luciferins and luciferases, so the substrate molecule and the enzyme that catalyzes the reaction, from all 4 of those different lineages that we knew as being bioluminescent at the time. So he had 1 species from each lineage. So here's Armillaria mellia, Neonothopanus gardneri. This is that Geronema viridolucens, Mycena luxaturna. And then this one, Bilobiletus gracilis, this was our control. It's a non-luminescent species. And so what Anderson did was he did cross reactions. He would extract luciferin from Armillaria, and he would mix it with the luciferase from Neonothopanus or Geronema or Mycena luxaturna or our control. And what he was able to show that no matter how you did these crosses, whether you had the substrate or the enzyme or vice versa, anytime you combined them from these separate lineages, they produced measurable light. When you combined them with the same extracts from this non-luminescent control species, no significant light produced. Okay? This was really groundbreaking for us because this sort of confirmed what we had always been hypothesizing, is that all 4 lineages share the same exact substrate molecule and the same exact enzyme that's catalyzing that reaction. And nowadays, of course, we know, because we've now elucidated the entire biochemical pathway, that indeed that luciferin and those luciferases are the same for all bioluminescent species of fungi. All right, and so just to wrap it all up, what our data is telling us is that bioluminescence evolved a single time in fungi. And that's that little green dot I had on the tree that I showed you initially. So this right here, looking at where these 5 evolutionary lineages occur, that is where the earliest in the evolution of this group where bioluminescence could have occurred. And this has now been verified by going in and looking at the entire genomes of a number of species and seeing that the genes responsible for the pathway, those precursor molecules and the precursor, the genes that produce those molecules were actually, look like they were probably present that far back in evolutionary time for these fungi. But what it also tells us, of course, is that it's been lost multiple times. So if we go and look at any one of these 5 evolutionary lineages, they're not 100% bioluminescent. Something like Mycena, 96 out of probably several thousand species are known to be bioluminescent, but there's probably thousands of non-luminescent Mycena species out there. And the same thing can be said for all these other lineages. So undoubtedly, probably through gene losses and various mutations that have rendered genes non-functional, many species have simply lost the ability to bioluminesce over evolutionary time. But remember, I told you at the beginning to look at here and notice that there's no green anyplace else in the tree. And what's really amazing about this is that it only requires a single loss of that biochemical pathway, the genes responsible for it, in this part of the tree here. So here we have the origin in green. If we had another red dot right there, that could be the loss of bioluminescence, which would render everything else within the Agaricales non-luminescent. Right? I think it's pretty cool. All right, folks, with that, I'll say thanks for your attention. I want to thank folks, of course, for specimens. So lots of folks have played a role in this project. I wish all the photographs were mine, but they're not. So lots of folks are owed credit for those. And then, of course, our funding to do this research over the last 17 years or so. Thanks. Happy to take questions. Yes, I can actually see the audience. That'd be great. All right. There you all are. Yeah. Yep. So metabolically, they're remaining metabolically active, right? So to produce all those enzymes and to keep that cycle going does take energy. So NADPH is involved. Is it energetically, does it cost the organism to produce the light? So metabolically, is it expensive? And the answer is yes, it does cost them something. Yep, certainly. And one of the things I always really want to point out is, we look at this and we're talking about this as if this is, when we think of evolution, we think of natural selection favoring a trait or favoring a behavior or something, and it's going to increase the frequency of that, over evolutionary time. I just told you that these things represent less than 0.005% of all fungi are bioluminescent, which I say suggests that this may not be massively advantageous. This may actually just be evolutionary baggage for these things, is another way to look at it. Yep. Else? Yes. Mm-hmm. That's a Phylloboletus manipularis. Yeah. How it got there? It probably came in with plants. Either someone brought something in. I don't think it floated there. The currents don't explain how it would get there that well. But my feeling is probably human introduced would be the most logical explanation. It could be landscaping. One of my favorite sort of hypotheses for fungal dispersal is mud on birds' feet. And that mud contains fungal spores or fungal propagules, and then that bird flies someplace. That's a long way to go from the closest area where we know it occurs all the way to Hawaii, but it does occur, or it does, that does happen. But my mind would go on human introduction for sure. Could have come in on someone's shoes. It could have been, someone might have been down in, I don't know, in Micronesia, hiking around, and then, came home with their hiking boots, which had mud on them, and, knocked all the mud off them outside their house, and sure enough, now you got the fungus there. Yes, yes, in the back. Yeah, so National Science Foundation, that was the grant. So when I was finishing up my PhD, I applied to the National Science Foundation for an award to do the systematics and evolution of the mycenoid fungi. And so as part of that project, that's actually what a lot of that research was done as part of my postdoctoral work, which was all funded for and paid for by the National Science Foundation. These ones here, all of these that you're seeing, this one and this one are my Brazilian collaborator. So this is one that is through FAPESP, is the university that he's at in Sao Paulo. This is the federal university there. So this is just each state in Sao Paulo gets money from the government to do biological and other types of scientific research. And so he just applied for funds from them. So in addition to doing the biochemistry, they're probably also, doing engineering and biology and all sorts of other types of research. Similar thing down here. This is a Brazilian Council for Scientific and Technological Development. And then this one was always shocking to me, is that the Office of Naval Research is very prevalent in Brazil. And they're in Sao Paulo, and we just happened to get to know someone that worked with them, told them what we were studying down there, and they encouraged us to apply for funding, and they gave us money. So yeah, actually a lot of this money from these folks actually paid for me and Desjardins to be able to go down and do multiple trips to collect mushrooms in Brazil. Yeah, yeah. Try and get money from any place we can. Yeah.
I noticed that pyruvic acid was like that.
Yep, it's in the pathway. Yep.
And so if the acid would be doing it with like the process is like what the process is.
Yeah, and it certainly could be. So it's playing a role in recycling, basically getting luciferin back to its original state so it can then be catalyzed. So yeah, absolutely. I mean, that's playing a role in the pathway. I don't do the biochemical work. That's totally beyond my biology knowledge. But what my collaborators have all, discovered working on that system is that it's caffeic acid is typically the limiting factor. And so I was telling folks, if you're growing, if you order, Panella stypticus or something like that from some culture supply company, and you're growing it on rye grain, it probably won't luminesce because rye grain is incredibly poor in caffeic acid. If you supplement it with caffeic acid, it'll start glowing. It is, yeah. So you can order it. It's not not hard to get. Yeah. So if you're trying to grow this stuff at home and it's not luminescing, chances are it's a caffeic acid issue. More than likely. Yep. Yes. It looks a lot like it because it's a cyclic pathway, right? And this is all close. Yep. Yeah, no, and it's all very closely tied to, I mean, it's highly and closely tied to the basic metabolic pathways of these fungi. So as these fungi are breaking down cellulose and breaking down lignin, they're generating these reactive oxygen species in the cytoplasm, which can be damaging to the cellular components. And so these fungi are more than likely producing light, metabolically expensive to produce, but obviously worth it for them. They're investing energy to have this other biochemical pathway going on, which is going to neutralize those reactive oxygen species, so acting in an antioxidant fashion. But you're right, being anything cyclic like that, respiration. Yep. That I don't know if we know that much yet. Yeah, good question. I'm not sure. Yep. All the same. Yeah, it's the chemical reaction. I mean, that's kind of the first hint we had that they were all using the exact same substrate molecules and enzymes, was that the light we measured from all of these different species and just observed by looking at it was all the same. So I think it's just the fact that it evolved a single time. And so it's always putting out, light at that same exact wavelength, or within that range, I should say. Yeah.
Is it possible in the laboratory test when they were testing that chemical, were they able to get the light, the wavelengths outside of the typical range?
No.
Or is that chemical reaction stuck producing that specific wavelength?
It seems like it's stuck producing that wavelength, and they can measure it. So you just saw that it's a range of like 520 to 530. So it's always, somewhere in that general range. We've never seen it higher or lower than that in a natural system. Yeah, and even in all the cross reactions, it's always the same. Yeah. So someone else had one. Yep.
In the like families or just in Jesus, my question is that within there's some like likenized species of fungi.
Mhm.
Have you seen, are there any bioluminescent lichen? Have you ever seen that?
Yeah. So we've not seen any bioluminescent lichens, and none of the lichenized basidiomycetes are luminescent. So even though they're in some of these families that are very close, None of them have, at least yet, been observed to produce any bioluminescence. Yeah, so it seems like that pathway was lost from their ancestors. Yeah, yeah, in the back. You mean this one? So as far as in regards to where the luminescence is? Oh, I see what you're talking about. Yeah, so that's the way. So yeah. Yeah. So the tree, this is the root down here. And so this, like, if this tree were not circular, if I uncoiled it, this part down here would be like at the bottom, and all these other branches would be going up towards the roof. And so it's just the way the tree is drawn. It's drawn in sort of like a ladder-like fashion. So each one of these branches, this is basically, you're going further back. Sorry, my pointer's dying here. You're going further back in evolutionary time as you sort of reach back. So all this that you're seeing here is swirling closer in towards the middle of the tree. And that's just the nature of the way the branching diagram works. I think I understand what you mean. Yes. So the other thing that we're doing here, when we make trees like this, we root them. So see this line here? Come back so you can hear me. This is the root. So I've actually told the analysis to root the tree. If we didn't have any root and this tree was just like, it would just be a big, star sort of pattern. But we know that I use the Bolitales here to root the tree because we know that they're the sister lineage, or one of the sister lineages, to the Agaricales. So I'm telling it to draw the tree in a sense that the Bolitales are the earliest diverging group. And that's why the tree sort of has this spiraling out sort of look to it. Hopefully that makes sense. But yeah, that's just the way the tree is drawn because of the out group rooting that we use. Yep.
Like a rough time period estimate for like the degree dot, like bioluminescence extent potentially.
Yep. So there was a paper that came out. I didn't bother doing it in our review here just because it's kind of been done. Calibrating trees is wonky at best, right? So we're using fossils. Fossils, turns out Basidiomycetes are really poor when it comes to fossils. There's not many out there. So we have a couple like mushrooms and amber and things like that we can use the data tree. Based on full genome level phylogenies of about 60 of these species of Basidiomycetes, it looks like the molecules that are required for this, at least the luciferin, about 165 million years ago, it looks like the gene family from which luciferase comes from appears about that. That's the date we would assign to it. Mm-hmm.
Is there any effects
That's what we've been very curious about that. So, and this is as we start sequencing more and more genomes, we go through. So every time a new genome is released, we actually go through and we start looking for luciferase genes and the genes for the other enzymes, histidine synthetase and things like that, and they're not there, right? And so they're lacking from a lot of these things. But I would be really curious to know if other things that are in these lineages, if you gave them caffeic acid, would they luminesce? So I've always joked that I'm going to get a spray bottle with caffeic acid, just walk around the woods and like squirt every mycena I see to see if it starts glowing.
Or is there any negative effects? Like this caffeic acid harmful
Mm.
in any way? Like is it stuck up? Is there any negative?
Yeah, not that we know. I mean, like caffeic acid is an incredibly common and widespread plant metabolite. It's all over. So these firefly petunias that you can order now, they're just utilizing the caffeic acid that's being naturally produced by the petunia. And so you insert the fungal genes responsible for the biochemical pathway here, you insert that into the genome of the petunia is already making tons of caffeic acid. So as long as that plant is healthy and happy, is getting all the water and sunlight that it needs, it just continues to luminesce. It's really crazy.
It's crazy. That's what the caffeic acid is.
Yeah,
And
somehow.
like,
Yeah.
if there's some negative aspects.
That I don't know. Good question though. Yep. Mm-hmm.
So you sound things about how the kind of like stuff like being fungi. How does that translate over? Is it translated over from the last one ancestor, or like why plants produce it and fungi produce it?
So
I mean, I know it's through some, but
it's so no fungi produce caffeic acid.
oh, okay.
They're getting the caffeic acid that they're using in the pathway is coming from the substrate that they are eating.
Okay.
Yep. So it's coming from the environment. Yeah. So it's a biological that they're getting. Yeah. Huh?
That's a really good question, but when I look at this thing, is it possible that the evolutionary advantage of having this disappear? So this is just residual.
Yep. Absolutely. So as I said, to me, it could be evolutionary baggage. It could be that, going back, and so we know when the genes, at least some of the genes that are required for the pathway, they evolved about 165 million years ago. So you can imagine that, many of the fungi in these lineages at that time, ancestor was probably bioluminescent. And then over time, as mutations accumulate, what we saw is that, species would just lose the ability to bioluminesce. Certainly doesn't seem that it hampered them, right? So if bioluminescence were what we call a key innovation, then we would expect to see adaptive radiations that surround it. We don't really see that in most of these lineages, with the exception of Mycena, right? So Mycena, there's 96 species within the mycenoid lineage that are known to be bioluminescent. Mycenas are one of the most diverse groups of saprotrophic mushrooms on the planet. So we could make the argument that, bioluminescence could be a key innovation that led to, incredible diversification within that mycenoid lineage. But there's not enough data to suggest it, because when we look at the existing species today, it's only, this here 96 out of probably a couple thousand species that are known to be luminescent. So it's hard to make that argument that it's super beneficial. So my thoughts is that it's been lost a number of times without really hampering the species in which it occurs. And it could be that in those lineages where it still persists, maybe it serves some other purpose than simply being a metabolic byproduct. So maybe in those lineages, it really has been selected to act as an aid for nocturnal spore dispersal, or attracting predatory insects to come eat the fungivorous insects or something else like that. Really, really hard to test, and hard to prove that kind of stuff. Yep.
There's no way to go with those.
Oh, as far as like losing it, over time, I would, things are both going extinct and evolving, right? So we're seeing that these lineages are, they're still continuing to diversify, but they're also still going extinct. We have extinction rates and diversification rates. Usually they pretty closely track one another. So we're not seeing a contraction or an expansion of bioluminescence in these lineages right now. At least not with the tools we have available to us. Yep. Yes, in the back. No. So what we see looking at the tree here is that these are all white spored groups that we see. So all of these species that are currently in these lineages, in the old days, we treated all of them in the family Tricholometacea, which was this large family of white spored agarics. So yeah, it just so happens that because of where it originated and persisted, it's all in these white spored groups. Yep. Yes, one more. Yeah, so in addition to inserting the biochemical pathway into, it's been put into yeasts already. So my collaborators' lab, they transformed it into yeast that you can use. So when you're doing biofuel productions, oftentimes you will use a yeast to initiate the breakdown of cellulose. And you can basically use a bioluminescent yeast species so that when you're doing it, you can ensure that you have the proper species that you've inoculated your biofuel substrate with. So that's one thing that was done early on. Right now, that company is really, they're putting the bioluminescent gene cluster into more and more plants. So we are going to see more and more house plants and garden plants coming on the market that are bioluminescent. So soon you will have a whole yard filled with glowing plants. Sorry? In theory, yes, right? And so what the companies do though, like I have the firefly petunia at home and it produces all sorts of flowers, but guess what? Those flowers don't set seed. So you can't collect the seeds and then grow more. So typically what they do is they're selecting these plants and they're modifying them so that they don't set seed that allows it to spread. But we'll see. There's a interesting company in Northern Europe that's trying to develop energy-free light that is based on the bioluminescent pathway. So they're really looking at this as trying to, create energy-free sources of light that can be used. So that's kind of where I would like to see most of this go. I don't know if the potential is there. It doesn't, and the light could be pretty faint, but it's really, I'm intrigued to see where it goes. But I'm sure it'll end up in lots of different places as reporter markers and things like that. All right, thanks everybody. Pleasure.
So I know it's getting kind of late, but I have one little surprise if anybody's interested.